Welcome everyone to this OTS webinar. Thanks for joining us. And my name is Alex Garanto for those that do not know me. And also we have here in the call moderating Dan O'Reilly that you are probably also know from the OTS. And today we have the honor to have as a speaker, Virginia Arechavala-Gomeza from Spain. So a little bit about Virginia before we move to her presentation. She is a trained pharmacist that did her studies at the Basque Country in Spain. And then she moved to the UK, where she did her master's, her PhD, and also the postdoc. And during this time, she participated in the first two clinical trials for eteplirsen and the preclinical development of these and other antisense oligonucleotides that are currently approved for Duchenne muscular dystrophy. In 2013, she moved back to Spain with a Miguel Servet and a Marie Curie Career Integration Grant. And since then, she is the group leader of the neuromuscular disorders at the Biocruces Vizcaya Health Research Institute in Bilbao, well, close to Bilbao, center of the universe. Very important also. In addition, she is the chair of an international network called DARPER, that means Delivery of Antisense RNA Therapeutics, where we aim as a European group and some international partners to improve antisense technology. And today she's going to tell us a little bit more about her experience with eteplirsen and how all these clinical trials were done. So Virginia, the floor is yours. So if you can share the screen.
Okay. Can you see it now?
Yes.
Perfect.
Perfect.
Okay. So, hey. Perfect. So thank you very much to all of you that join us today. Just a warning that today is going to be a pretty simple lecture. So it's not going to be any high level chemistry or anything like that. So I'm just going to take all the glamour out of developing an orphan drug. So first of all, wait, it's not working. Okay.
Yeah, we see.
So is it working now? Okay. First of all, I'm just going to give you a tiny bit of background on Duchenne muscular dystrophy so you can follow through, and then I'm going to tell you a story. And that's going to be it for today. So I'll begin at the beginning and then I'll stop. So first of all, in the introduction, Alex already did that for me. So I did my first degree in Spain, and then I moved to the UK for my PhD and postdoctoral time. And now I came back home to Spain, to Biocruces Vizcaya, where I work as an ICERVAS research professor. So on Duchenne muscular dystrophy, which is the little bit of background that I'm going to give you, just as you should know that it's a rare disease. Many of you probably are quite aware that rare diseases are defined by how common they are and not how weird they are, as they are in Spain. And Duchenne muscular dystrophy is one of those. It affects 1 in 4,000 males, so it has a worldwide prevalence around 300,000, and is caused by mutations in the DMD gene. What you get is a progressive loss of muscle mass. So there is a degeneration and regeneration of muscle mass, and that muscle is replaced by fat and connective tissue. So what happens in this disorder is that over the years, you lose the function of those muscles, and then you lose some functionality. So you lose standing, ambulation, self-feeding, and usually death comes around, in their 20s or 30s. So the reason for this is this loss of muscle mass. So you see muscle at the early stages is a bit more normal, and then you see how you go losing that, and it gets replaced by fat and fibrotic tissue, and by the end of it you only get a handful of fibers there left. This muscle is lost because there is one particular protein that is missing, which is dystrophin, and this protein here is part of a big jigsaw of proteins that link somehow the inside of the cell and the outside of the cell. So that will be dystrophin here, binding the actin cytoskeleton, and through connection with many other proteins binding to collagen outside. So it's a little bit, in a way, a structural role and a mechanical role, although it has a few other roles, but that is the main function of dystrophin, and that's why it's a bit the Achilles heel of skeletal muscle. So when the protein is missing, there is an increased permeability and membrane instability, so then you lose that muscle. So you could consider that a bit like those, springs in the trampoline, that you may not last very long if you're jumping over that. The other thing that you need to know is that dystrophin is huge. It's huge as a protein. You can compare it here with other standard proteins, and it's also huge as a gene. And so that also involves quite a lot of trouble when you're trying to work with it. And the other thing that is important is that the deletions and mutations of this protein are not just one particular deletion, but you may have a whole exon missing or several of those in different areas, larger, shorter, there are duplications, replications. So there's quite a lot of different mutations, which means that you're going to need to make personalized medicine, and you're not going to be able to fix all patients want one single treatment. If you've seen Duchenne muscular dystrophy, which is a fatal disorder with a slow but quite quick fatal disorder, you will be surprised to see a very similar disorder, Becker muscular dystrophy, which has a mean age of onset, which is much later. It has a very variable progression. There are patients that never have any symptoms. Only some of them lose their walking ability. And regardless of all of that, the cause are mutations in the DMD gene. So I've just told you that you can pretty much change that gene anywhere, and you'll get Duchenne muscular dystrophy. And yet you see those patients in which you do have mutations in that very same gene, and you have a much milder disorder. And the reason for that is something that you may understand when you see the exon and intron types. You see that we have conceptually this idea that the codons are going to be very nicely ordered when you have the introns. You have an exon here and then an intron and the next codon, but actually that's not how it happens. You may have them interrupted at different places. So if your codon is interrupted here, it wouldn't fit with this other codon there. So this is the main reason. And when you see the dystrophin exon, you'll see that there are many areas in which this will probably break the codon. Is the reason why the open reading frame may be disrupted when you have deletion of different exons. So, for example, you may have a deletion of exon 50. When splicing happens, you have a disruption of the open reading frame, and you don't have dystrophin. Whereas when you have a deletion that it might be even larger, but it still keeps the open reading frame, you will have the open reading frame maintained, and you will get a shorter but functional protein. So that is the basic that you need to know, and then I'll go to the story. That's the story part of today's webinar. So first of all, a disclaimer. I'm just going to tell you the way I saw it. So it might be completely different to what my supervisor thought of that, and it may be different to what I may tell you about it in, I don't know, let's say a couple of years. So the story starts with a consortium of different investigators from many different universities that got together to test something quite new. There were people mostly in the UK, but also people in Australia, and people collaborating also in the Netherlands. And there were 3 different patient associations. And that is quite important because they managed to get funding from the Department of Health to be able to start a clinical trial. So I joined as a postdoc on that grant, and I was employed by Francesco Montoni and Jennifer Morgan, who were my supervisors at the time, at Imperial College London. And we were going to do something that at the time sounded very exciting, which was exon skipping, and we were going to use something that at least I hadn't heard about yet, which was antisense oligonucleotides, and it was going to be personalized medicine. That all looked great, but actually we had absolutely no idea, or I had absolutely no idea how it's going to be. So this is an example that I use for students. So I'm sorry if for many of you that will be a bit too simple. But when we consider our DNA as this large library and our genes of these, our genes being that recipe of that book, we would have this quick copy of our recipe that will be the pre-messenger RNA, and then we have that cleanup. So then we have a complete recipe, just a complete set of instructions that will tell us exactly how we need to cook it. We take it to our ribosomes or cook, and we cook. And we have tons of different nucleic acid therapeutics, and there are many ways in which you can alter that process, and this is the beauty of it. So this is an example that is probably quite well suited for Spaniards, but I think many of you may also recognize Jamie Oliver. And the thing is, a few years back, he did publish a book on Spanish food, and he made a horrible crime for some people in Spain, which is to include chorizo in his recipe. That is a horrible, terrible mutation that will cause death to people from Valencia. So if you do have a recipe such as Jamie's, it will cause a disease. In our case, what we will need to do is, if we do have that book, that, bad luck, you managed to get Jamie's copy for paella, you need to take advantage of this. So you include a post-it saying no chorizo, please. And then once that is cleaned up and you get the instructions, then the recipe will not have the chorizo and you'll have a proper paella. So problem solved. So that was what we were trying to do basically with exon skipping. That's how I understood it at the time, because I didn't have no idea of what we were doing. So we would get our post-it, our short sequence of nucleotides, to hide the chorizo, to hide exon 51, and put it back in frame. So that would be the idea of making a bit like Becker-like protein, if you want to see it like that. So the first thing that you need to do is to make sure that this works. And the idea is behind this was that we were going to get to a clinical trial. So for that, we needed a sequence. We needed a drug that we can include in the clinical trial. So the first thing that you need to do is to prove it in a model, and in this case was samples from patients. And because dystrophin needs to be, is the gene that we're interested in, and it's not expressed in just anywhere, we needed samples that were muscle cells. You can also do it in fibroblast, but it's either fibroblast or myoblast from patients with the specific deletions. When you start looking into something like that, you need samples, and those samples are in biobanks. The problem was that at the time, even though we were in the largest neuromuscular center in the United Kingdom and probably one of the largest in the world, when we went checking for those samples, we could only find one in our liquid nitrogen, because those need to be collected and cultured and kept to be able to be accessible. And for that, you need biobanks. After searching high and low, we managed to find just one other sample. And that was in a biobank in Germany that we borrowed. And then we made all the experiments that we needed to do with those two samples, which meant that we couldn't do as many experiments as we would like to do. So what we did was to do a first round in control cells, and then some of those in patient cells. Probably if we had done this today, we would have tested many more molecules, because we would have been able to do more stuff. So that's one of the very first things, biobanks. Those are important for any work in rare disorders. So this is what we did to select the best sequence. So the idea was that there were already quite a few groups that had designed very good sequences to skip exon 51. So what we did was to collect those and compare those blindly on the cells that we had. So that's what I did. I just collected those from Leiden and from Australia, from Royal Holloway, and we compared those. And then we had the best two, which ended up being eventually disaprelsen and eteplirsen, which we thought were the best. We showed that they were the best at making exon skipping in patient cells and restoring dystrophin expression in cell culture. So I'm going to tell you the story of mostly of eteplirsen, but actually disaprelsen, which is this over here, went pretty much around the same path, a little bit ahead of us actually, because it started before. So the other thing that you need to know about those two drugs that went on clinical trials is that they had different chemistries. So one of them had 2-O-methyl and phosphorothioate backbone, and the other one had PMO, a morpholino backbone. And that is important because it compared both drugs' different pharmacokinetics and different capabilities. So it's a bit like having a plaster, that it could be a plaster of fabric or a plastic of plastic. I'm actually allergic to the adherine in one of those. So that does make a difference to patients, and it does make a difference as you see for these kind of drugs. If you see, both eteplirsen and disaprelsen were extremely similar. This sequence here is this sequence here. So actually eteplirsen is just a little bit longer than eteplirsen, but includes the same sequence. But because they had different chemistries, one was administered subcutaneously and the other one was intravenous, one of them did bind serum proteins and had a very long half-life, whereas the other one didn't and had a really, really short half-life. So it was a very extremely expensive urine. One of them did have some adverse events with proteinuria and a very strong local reaction, whereas the other one didn't have any. So both of them went into clinical trials, and that is, the usual way in which you compare all the different drugs. So the first thing that you need to do when you're doing a clinical trial is to be trial ready, and that is an interesting concept because you need to know where those patients are and who would take part, and there's going to be some different reasons to include them or exclude them for a clinical trial. And actually, it's not that easy to know where the patients are and to have those patients ready to go. And for that, a huge part are registries. Registries is something that are not often very well kept, particularly for rare disorders, and usually is something that mainly patient associations are more responsible for. So that is something that will probably change a lot the speed at which you may be able to organize a clinical trial for a rare disease. So if you don't have that ready, you'll probably not be such a happy chap after a while. So what we had to do is to get ready. We were going to inject a very, very small muscle on the foot. So first thing that we had to do is to check that muscle was still there, because actually some patients didn't even have that muscle. That will be the extension digitorum brevis, who is a foot muscle. And the first clinical trial we were going to do was going to be on patients that were already wheelchair bound. So we would inject one of those with the drug and the other foot with a saline. So we collected samples, we got ready for that. And we also said, okay, we need to make sure how we're going to evaluate these clinical trials. And the thing that we didn't know is how efficient this was going to be, how it was going to work. We hadn't done this in humans yet. So we said, okay, we need a way to measure this. And obviously we wanted to express a protein again. And the first thing that anybody thinks about is Western blot. But we were going to have tiny samples, and Western blot for dystrophin is a mess. Dystrophin is a huge protein, as I just told you. To be able to see it, you need to load a lot of sample in the gel. So you're going to be wasting a lot of your very precious sample from a very sick patient in a rare disease in a once-in-a-lifetime clinical trial, and you don't want to use it all up in one single experiment. So what we were doing while we were getting ready for this clinical trial was also to prepare for that and develop a method of immunohistochemistry. So the method that we developed was able to check the intensity that we had in those sections. So a section is just a very small amount of that biopsy that we get from the patient, and we get the corresponding intensity readings. So with that, we develop a method to quantify that intensity, so then we could get ready for whatever we will see in the clinical trial. So then we went and we injected those patients, and these are the results of the very first clinical trial. And you can see here on top the untreated contralateral foot, and this is the treated foot. So that was great. Actually, it's one of those things in which you are in the lab, and I was checking those samples, and I was blind to them, and no one had to tell me which one's the treated one and which one wasn't. So that was actually great, because you could see very easily that it had worked. So that was a very good result. We were very happy with this. And also, we were happy that we were able to use the method that we had developed to evaluate it. Actually, it took a while to get it published. Actually, the clinical trial came out in 2009, and this was published probably a year later. And actually, a couple of years later, there was a slightly modified method from ours that was published in the very same journal, which was, interesting. And then we saw that someone had made a review on that other method, and we were like, oh, what do you mean? We published this first, they do it later. So we got a little bit, not angry, but we decided to send a letter saying, hey, our method is perhaps a bit better. And then we thought about it, and it was a bit silly to start thinking about, whose method was better, because at the end of the day, what you want to do is to measure it and to make sure that you can all measure it the same way. So what we did was to start thinking about how to collaborate. And also, by then, there was yet another method that was much more expensive, because it required a much more expensive equipment. So we decided to collaborate so then we can all do it together and do it the same way. Because if you are going to test a drug for, let's say, high cholesterol, anyone can measure cholesterol anywhere in the world, and they will do it the exact same way. So it will be very easy to check how your drug is working. But if you're doing dystrophin, we each had a way of measuring it. So we needed a way to make sure that we were all doing it equally. So we collaborated to compare the methods, and then we saw that it was possible to make a standardized, validated method, which was very good, because that way we knew that we could be more assured of what we were measuring. In the meantime, because we had a very good first clinical trial, we started with a second clinical trial and with quite a lot of other stuff. This was the first patient that took part in the second clinical trial. And we saw results. They were not as good as with the first one. Here, those are the pre-treated and the post. So for some patients were quite good, but for others were just a little bit more faint. But yet, we saw that the drug that we were giving those children, in this time, it was not local, it was an intravenous drug. And then we saw that we could see exon skipping and that we could see a bit of restoration of dystrophin, which was good. So we said, okay, we'll celebrate it. We got the paper accepted in Lancet. So we were ready for the press release, and we said, okay, Mom, I'm going to be famous for like 10 minutes, so make sure you buy the newspaper. That press release was going to come out on Monday, and this happened over the weekend. So we never got our 15 minutes of fame, because there were far other more important things to talk about that week. But yet we managed to get those results. And with those results, then you need to go further, because we had only a phase 1/2 clinical trial in which we had shown first that it was safe, which is the main thing when you're doing a clinical trial, particularly in children and with something that hadn't been tested that often. And then that there was a little bit of efficacy, but you needed to go further to show, a good dose, a good result. And that had to go further into more patients in another clinical trial. And the regulatory agencies, which are the ones that are going to approve your drug at the end of the day, will want something, safety. They like placebo-controlled trials, and they want clinical outcome measures and biochemical outcome measures. I've just told you about biomarkers and dystrophin. That will be a biochemical outcome measures, but there are also the clinical ones, which are far more important because those tell you if your patient is taking any advantage from your drug. So the thing with the natural history of the disease is that you need to know what is going to happen if you do nothing. And with those patients, not many people knew. People knew that the patients were going to get worse, but it was difficult to measure how much worse. And you need a number, you need a way to say, okay, this is slowing down the progression or not. And the way to measure it was decided to be the 6-minute walk test. So you can see here in the picture, 6-minute walk test. So basically you have, the distance, you have someone with a stopwatch, and you walk for 6 minutes. So then you know how many meters you've covered. And one day you may do 600 meters, and then the next visit you may do 550. So then you know that you're declining in your 6-minute walk test. The thing is, there was no natural history data when these clinical trials started. So all those happened after they started. And when they were done, and actually these are some results from 2014, and I just told you that the first clinical trials for eteplirsen started in 2009, you see that there is a decline. You see here, from the amount that they walk on the very first visit, on the different visits, they will decline in the number of meters they were able to walk. And actually, they saw a couple of interesting facts. One is that there was a variability, and also that the age of the patients was important. So patients under 7 years old, they were still growing up. So maybe they were doing better, not because of your drug, but because they were growing. They will still be able to walk a little bit more. The starting value, how much they were walking on the first visit, is also important because there is a point of no return. So then when a patient is walking already under 350 meters, it's very likely that that person is going to stop walking altogether soon. And also the willingness, those patients want to do good. The patients want to do good. So they may train, they may have a better day or a bad day, or they may be more tired. So sometimes, they did a test, they offered them $50, and they all did better than in the previous time. So actually the willingness to take part is also important, and this also needs to be considered for future trials. Obviously, for longer periods and for the future, this only applies for patients that can walk, and there will be many Duchenne muscular dystrophy patients that are already on a wheelchair. So you need different outcome measures, and that is something also that you need to consider. The other thing that happened is that there was a biotech battle. So at first there was Prosensa and Avia Biopharma, which were in charge of those two clinical trials. And as you can see, the sequences were very, very similar. So they had some qualms between each other. Prosensa at one point partnered with GlaxoSmithKline, and they did a first clinical trial. The results were not as good as they expected, and Glaxo decided to pull out. Later on, BioMarin bought Prosensa, and they continued with the development of the drug. And Avia Biopharma wasn't bought by anybody, but they changed names. So that was Sarepta. It's Sarepta Therapeutics today. This trouble with the sequence meant that they started suing each other, even though the academic groups were quite good at sharing data and collaborating. Companies are not always that keen. So the clinical trials moved to the States. And there was a point when there was this thing in the media about a biotech battle and who was going to go first and who was going to be able to get a drug out for Duchenne muscular dystrophy first. I think it would be good to get one at least for patients, but that's the way it was portrayed in the media. And There was a point when there were many different clinical trials being reported, and as I told you, eteplirsen was a little bit ahead and also had a larger company behind. So they had some problems with the adverse events, and they had some bad feedback from the European Medicine Agency and from the FDA, and they decided to stop altogether all their work on eteplirsen and all the drugs of that generation that were being developed, which was a big blow, because you may think that suddenly there is just one in the race, but actually the whole field suffered from those, and there were quite a lot of people that were thinking that perhaps the whole concept of exon skipping was not worth pursuing. And there were very difficult times for the companies that were in the field. So the first of the trials that were done in the US was a dose-finding trial in which they had a few patients that were placebo for only a little fraction of the time, and then they were treated for a few weeks. So here you can see the pre-treatment biopsy, 12 weeks, and here you have longer time and 48 weeks. So you can see there is a restoration of dystrophin protein, but actually there's not a lot of dystrophin in there. But with those results, they went to the FDA for approval. The thing is, as I told you, there was no placebo control, which is not the best thing when you're going to get any approvals. And what they did instead was to compare the patients with the natural history. Again, the natural history are not exactly a good control. I mean, they got the patients from those patients had been treated in the US, and the natural history data were collected in Belgium and in Italy, and they got from a pool of data, they selected those that were using the same steroids, that were similar age, that were exon skipping amenable, and that they had the similar deletions. So they had those patients here, 13 patients that they compare with the 12 that had been treated. Yes, they had similar deletions, they have similar treatment of corticosteroids, etc., but that is not quite the control. Still, when you compare those, the eteplirsen group with external control, you do see a difference between them. Although it's not a huge difference, but you do see a difference. Both of them keep on declining. None of them is still, this is the 6-minute walk test, and there is a decline. But you can see a steeper decline in those that are the natural history patients. And here you have the loss of ambulation. You have here in eteplirsen two patients that stop walking on the very first year. Those probably were under the 350 meters, so they were already quite sick when they started the clinical trial. And you can see here that obviously those two patients never went back to walking. But you can see the amount of patients in the other groups increasing, while for those being treated, they don't. Again, it's a very small number of patients, so it's very difficult to tell if those patients were the lucky ones that have a slower progression or not. So it's difficult to convince anyone with this data. Yet this was the data that was put forward the FDA, and it was sent for revision. At that time, obviously there is a huge controversy, and there is a movement to get patients more involved into the approval of drugs and being able to have a say on what is relevant to them, which is great. So one of the very first times that the FDA had more involvement in the way that the drug would be assessed was at eteplirsen. And the meeting in which they first had advisory committee meeting in which a group of experts will give an assessment of what the data they have in front of them, and they will tell the FDA, okay, we think this should be approved, or it shouldn't be approved, or we don't think is useful for the patient. This is this is a report of how that meeting went. That meeting was, they had to book a really huge room that was overflowing, and it was live on the internet. There were quite a lot of people checking that with quite a lot of interest. But the report talks about heart-wrenching advisory committee meeting. They implored the FDA. That is not quite what you would expect when you're trying to make a cold-blooded decision of, is this working or is it not working? You don't want to decide because of feeling sorry for someone. You want to decide because that really helps them. And actually, the advisory committee said no. They said, we don't agree. We don't think this drug works. And that's what they told the FDA. The FDA then had to decide. And it looked a bit gloom, actually. But eventually, the FDA said yes, and they did approve eteplirsen. It was an accelerated approval, and it was extremely controversial. So people said, that they had lowered the standards, that they shouldn't have approved it. And actually, yet, when they talk about drugs like, the recent approval for drugs for other disorders, such as Alzheimer's, whenever anyone talks about a rushed approval, they talk about eteplirsen. They may have the reason, they may not, but it's actually one of the examples of that is usually used of how something was approved in a way that was extremely controversial. So actually, since then, the FDA has approved a few other drugs that work on the same concept, which are working, they target different exons, exon 51, 53, and 45. But the European Medicine Agency hasn't approved any of those, so it's only the FDA. So the approval has been made on the basis of dystrophin expression. It took us quite a lot of time to develop a method to be able to quantify dystrophin, and actually that was the cause of this approval was that they were able to prove that yes, there was dystrophin there, but also they were able to say that there was not a lot of dystrophin there. So if that little amount of dystrophin is good enough, well, there's some reasons behind thinking that is not good enough. It's not enough, and you can think that the amount is much lower than what you see in Becker muscular dystrophy. Those patients that are doing much better than in Duchenne muscular dystrophy. They said, yes, okay, that may work. Maybe in higher doses will work better, but not what you just showed me. And this poorly designed trial may open the gates to other, not so good drugs. On the other side, you may think that we don't know how much dystrophin you need to get a benefit, that there are results in mice with very low expression saying, yes, this works. And they have a better phenotype than patients with you do see a better phenotype in patients with residual amounts of dystrophin. Still, because we are talking about a rare disease, we don't know how much is good enough. And this has been the basics on the two different decisions, one yes and one no. And I think both of them are right, because you probably can back both of the yes and no sides with good reasons. So I think we learned quite a lot while we were preparing for this about the importance of biobanks and registries and treatment standards and the natural history studies and the validation of outcome measures. And mostly, I think we learned about cooperation, how these things need to be done by lots of different groups. So this is a paper that I always like to show when I talk about cooperation, and this was written by all these people, and this is only a handful of those. And it does describe what we learned while we were preparing for those clinical trials. And those are some of the authors. This is, an English parent, Italian clinician, Dutch researcher, myself, Belgian clinician, Spanish parent, regulator from the European Medicine Agency. So you do need all those people to work together to be able to advance to where you need to go. If you think about other disorders, if you see spinal muscular atrophy, is also another disorder in which you are trying to restore the expression of protein, because there are mutations in the survival motor neuron gene, and the severity of the disease has to do with the amount of SMA protein. So the main difference with, well, not the main difference, but some of the big difference between Duchenne muscular dystrophy and spinal muscular atrophy is that patients in SMA never roll, never sit, never walk, and they usually die before the age 2. Whereas, the natural history of DMD is much lower. So in a sense, it's much easier to show some results with a patient that has a very extreme course of action. And the other difference is that both drugs are trying to do something very similar. They're all both altering the splicing of the RNA, but one of them is trying to do exclusion, the other one inclusion, but they're targeting different tissues. One is targeting muscle, the other one is targeting motor neurons. And the way they are targeted has to do also with the delivery system. Some are intravenous and some are intrathecal. And nusinersen works extremely well. So I think we've all been learning one from each other, and this, for example, learned quite a lot from the development of nusinersen. It was a tailor-made drug made in record time. If you have a look at how long it took to approve eteplirsen, it was around 11 years, and it was fast at the time. But if you check nusinersen, it was around 7 years. And it was because the clinical trial stopped because it was working so well. That is fantastic. But for milasen, it was under a year to develop this drug. And this is something that has changed the scenery quite a lot. This was something that was published not in a scientific journal, but in The Economist at the end of 2019. And they were talking about how personalized drugs will be something big that will happen in 2020. Well, I think other big things happen in 2020, but this also happened. Actually, the NLOREM Foundation is working on individualized therapies for ultra-rare diseases. You also have things like the Dutch Center for RNA Therapeutics that is also aiming to develop these drugs. There are now guidelines on how to prepare those individualized antisense oligonucleotide products by the FDA. And even though milasen didn't quite work for the patient in the long term, it did increase the lifetime, the expectancy of that patient. And now milasmam is working as in the advocacy for this development. There are other examples like in ALS patients. This is a patient that also was treated with a personalized drug. Again, this drug didn't save her, but it's being tested now for other patients. So the drug that was designed for her is now being evaluated for other patients with the same disease. This is something that we also need to make sure that we know about. Like, this is another patient, Valeria, that was injected with valeriasen, which was designed just for her and also used for another patient. And they both had some hydrocephalia. So there are, I think, a few things that we still don't know about, but all the work that it was done to develop this drug is helping other children. And actually, the parents of Valeria are now in another association trying to develop these drugs further. So you see, with some of the drugs, we had very similar mechanism of action, different tissues, and we saw that the different delivery systems made a huge difference in how well they work, because eteplirsen doesn't work as well as we would have liked it to do, whereas nusinersen works quite well. And actually, the target tissue makes a big difference. There are some tissues that are easier to target, and this is something that became quite evident over the time, and we decided to try to solve it when the way we solve things, collaborating. So this is pretty much the way I see my colleagues had the cost action that I chair, which is this is our normal identities. And basically what we are is a group of people from academia, clinicians, pharma, patients that have expertise on different things, like on different target or disease tissues, or we have expertise on nucleic acid chemistry or drug delivery. So the idea is that we will try to join together to solve a common problem. So from all the talk that I've done on rare diseases, I think the interesting thing is that this is now coming to pretty much everyone, and probably things like therapies that don't target very rare diseases and now are in the market may help opening the doors also for other rare diseases, the same as rare diseases open the door for not so rare diseases. So I think what we may expect in the future is like lots of changes and lots of unexpected things. We have lots of other chapters and spin-offs, but basically what we do need, apart from researchers, obviously, is everything else, because I don't think you just need a very good drug. You need everything else to align to be able to get to the finish line. So I think if you do any of this, you'll be able to contribute to get those drugs in the market. So that's it. I said that I'll begin at the beginning, I'll go on till the end. So this is us in Bilbao, and we are looking for pre-docs and postdocs. So do contact me if you're interested, and I'll just take any questions. Thank you.
Great talk, Virginia. I think we can all agree that that's fantastic work you're showing, and also that having patients involved in research is very important. There are a couple of questions, in fact 3. The first is from Brian. Can you comment on the results from PCR versus results from Western blot? Was there a good correlation? Any comments or suggestions for applying this ASO approach to a new target? So I guess there's a couple of questions in that.
Okay. Well, PCR will tell you if there's exon skipping, but obviously you would need to know that there is some protein there as well. I don't like Western blots. I have to admit that. I've done enough dystrophin Western blots to hate them with passion, and that's probably why I've been developing other methods to measure dystrophin. But I think as long as you do it with good controls, and it's interesting how they have evolved over the years. Before we used to just run, pre and post and get a whole Western blot with tons of patients, and now pretty much you get a control with different dilutions and then just the pre and the post. So you will use one gel for one patient because you do need to get so many controls to make sure you get it right. That tells you how difficult it is to evaluate it, and also you're comparing it with a control. And controls are also very different from each other, so it's always going to be relative. I don't like Westerns, but I think we need to show that there is dystrophin there.
Great. Next question is from Steve.
Hi,
is
Steve.
Is it true that the FDA AdCom voted against accelerated approval, but the committee was split 7-6? The vote for full approval was by 7-3, as 3 members abstained from voting as they thought the questions were inappropriate. When drizzled send was considered by the FDA, the vote was no by 13-0. That is a clear no. Well,
yeah, I think the problem with I guess he meant eteplirsen. The adverse events played a huge part on the decision of on eteplirsen, and I think that was one of the biggest problem with eteplirsen. There was no adverse events, and then the only questions were about the clinical benefit, yes or no, and it was very short time to know if it was if the clinical benefit was there already or not. I think it was a bit early probably, but it's true that some of the members of the advisory committee complained that the questions were not quite, that the questions could have been phrased differently, so then they could have said something different at that meeting.
Another question is, a few of your slides show, and this is from Mike, a few of your slides show N loss on exons 42 to 45. Is this the portion of the protein required for functional dystrophin?
You cannot, I mean, actually in some of the mini microdystrophins, some of them do have NOS and some don't. And there is controversy of how useful they're going to be for patients. So obviously NOS is necessary. So we don't know yet how much better will be those mini dystrophins that will have the NOS. I mean, I think dystrophin is huge. There are many areas that are not as vital, but obviously it needs to bind to the right proteins, both at the N and C terminals, but also NOS is probably quite important. But we don't know yet.
Yes, we have the final question from Jeff Milton. What do you think is the biggest challenge for oligonucleotide therapeutics for personalized approaches?
Lots. Only one. I think delivery is a big challenge, obviously, but it's not just that. I think price is a huge challenge. Manufacturing is not as much as it is, like for example, for gene therapy approaches. But I think if you had asked this a few years back, I would have thought that the convincing people that this is possible was part of the biggest challenge. But I think now that we've seen all the development of RNA vaccines and drugs, I think everybody's a bit more ready to try those things. I think for personalized approaches is finding something that you can evaluate quick enough, because you're only going to have a very slim chance of being able to prove it. So probably for the very first patients, you will need something that is going to be easy to show an effect. So it has to be something bad enough that you can restore, but not so bad that it will be already past the point of no return. So I think selecting the patients is going to be the most difficult bit for the first clinical trials.
There is one more question that came out in the chat from Zul. In the 2007 paper, experience testing A20 and B30 ASOs in vitro at concentration of 50 to 400 nanomolar, the ASO delivery was using transfection or gymnotic uptake.
At that time, we were using lipofectamine. Yeah.
Thanks. And I guess that's it for the questions.
I have one question. Yeah,
okay,
I
Alex,
have my raise hand. So basically for all those people that are not, well, first of all, thank you for the nice presentation. It was awesome. But for all those that do not work in the muscle. So you said that both the EMA and the FDA were right, but now after all these years, who eventually was more right than the other? Is eteplirsen really working or not? Was the FDA decision a good decision or the EMA? Yeah, I mean, how is the status right now?
I think it's very difficult to tell. I mean, there are a few patients that had been taking this for many years, and many of them are quite stable, but it's difficult to tell, when you're talking about very small n numbers, it's not as easy to tell. So I think it does help, but the amount that you get from it, it's very difficult to evaluate. I think it's only for the patients to tell how relevant is relevant. I think you also have to consider the cost and the fact that those patients need to, take, it's not a drug, it's not a one-off, it's a weekly, in most cases, weekly injection, and it's a very expensive drug. If we could take away the expense and everything else from the question, I would say yes, you have to approve it. It's giving, perhaps it's giving something, but I don't know. It will depend on the particulars of every family and the patients and how they see it. I don't have an answer. I wish I had. I mean, we worked on delivering eteplirsen. I wish I could tell, yes, we did, and it was great for everybody. I think it's not as good as we would have liked it to be, but still, whenever I check on YouTube and I see one of those patients that are going to the high school reunion, I'm quite happy to see that. So who knows?
And how did the patients, of course, because I think it might be devastating in this case for the European patients, seeing that they cannot have access to a potential treatment, while in the US they have. So how did they start, try to convince the EMA or the EMA, or I don't know, I mean, just out of curiosity, how, because if I would be the patient, I would be quite disappointed. So
yeah, well, some of them are able to take part in clinical trials, but only some of them, not that many. I think I would also be disappointed, because when you're a patient, you know, you just want, but perhaps we've been lucky to see that there are now quite a lot of other clinical trials in place that are not only based on the exon skipping. So for Duchenne muscular dystrophy, I think it's been a huge change. I started on this in 2005, and there was pretty much nothing. And now you'll see lots of different clinical trials, not just on, you know, nucleic acid therapeutics, such as, you know, exon skipping or gene therapy, but also other things that are treating the fibrosis, or so there are other drugs in the market, and we'll probably need a combination of many of those in the long term. So perhaps you can always think that, you know, they may be able to benefit from other drugs until we get, you know, eteplirsen 2.0 ready to go to help them, or a very good gene therapy. So I think at least we have other things to use in the meantime. But I don't think I can put myself in the place of patients, because I don't think I don't think I can give an opinion on what they may or may not feel.
thanks.
Great, thanks so much. I don't think there are any other questions. But I would like to announce that there is a December webinar by Bruno Godinho. So he's a great speaker, and his work on the divalent siRNAs is really cool. So I highly recommend joining in that. But I would like to just thank Virginia again for a great talk. And I won't be eating any Jamie Oliver paella. Don't worry about that.
Well, you should. I mean, I have something to say. I hope no Spaniards are listening. I have included some chorizo in paella many times before, and I bet Alex has also. It's just that if you are from Valencia, you know, people from that particular region in Spain, they're very picky about their paellas, and you know, I think it's good.
Okay.
Okay.
I don't think I ever put chorizo, but it's fine.
You haven't made paellas properly then.
Okay, then thank you very much to everyone for joining, and especially to Virginia for the very nice presentation. And see you on the 1st of December, as Dan already announced in the presentation of Bruno. Bye-bye.
Bye. Bye.